Question 5. Scientists investigated the hydrolysis of sucrose in growing plant cells by an enzyme called SPS. Name the products of the hydrolysis of sucrose. So this is the biological molecules topic, testing your knowledge of disaccharides. Sucrose is one of the examples of this. All the disaccharides that you learn about are made of glucose and one other molecule. In this case is also fructose. And that should give you the 2 marks there. We'll just have a look at it there. There we go. Okay, the next part. The scientists grew plant cells in a culture for 12 days. At the start, there are only a few cells. Then each day, they determined the mass of sucrose hydrolyzed by SPS in the plant cells in 1 hour, and they've given you the results. And they've asked you to calculate the rate per minute of this reaction, record these in standard form, and then plot a graph. So there's quite a lot to do here for just 3 marks. So I've already gone through and calculated the rate. All you had to do is divide these numbers by 60 minutes. And on your calculator, it should already do it in standard form for you. And then again, I've also done this bit because it took a little while to do, but I've plotted it on a graph here. Now, what's really important is that you get the axes the right way round. So it's time on the x-axis, and then we're measuring rate, which will be on the y-axis. And you need to make sure you put the units in there as well, and you're able to identify what they are. So because we've done micrograms per minute, we've divided that by minutes, so we do micrograms times by minute to the power of -1, and then we also have to times it by 10 to the -3 because all of our results are 10 to the -3, and you won't get the mark if you write on the y-axis 1 times 10 to the -3, 2 times 10 to the -3, and so on. They want you to include it in the units. And then, so I've just plotted them all there and joined them up kind of dot to dot. So what you actually get the marks for on this, we just put the mark scheme over. So you get the marks for the axes being labelled correctly, then with the correct units on the axes, and then actually correctly plotting them and doing... Okay, so what can you conclude about the growth of plant cells from these data? Explain how you reached your conclusions. So we've got 2 bits of data. We've got the raw data here and we've got our process data here. This one's probably going to be easier to look at. But remember, this data is talking about sucrose. It's talking about rate of reaction. So we need to infer things about growth from the rate of reaction catalysed by this SPS, the rate of hydrolysis of sucrose. So the first thing we need to do is establish a link between sucrose hydrolysis and growth. So how is sucrose hydrolysis linked to growth? Well, it might be that when sucrose is hydrolyzed, you release glucose and fructose, as we've established in the earlier part of the question. So perhaps this glucose can be then used for respiration to produce ATP, which is going to release energy for use in reactions to do with growth, such as cell division. So now we've said that when there's a high sucrose hydrolysis, we've got a high growth. But now we need to conclude something about the growth of these particular plant cells from the data. So let's have a look at the data. So we can see the rate of sucrose hydrolysis gradually increases, then increases quicker, but then levels off. So what we can infer from that is up to this point at 10 days, we're having growth, we're having more growth. Then after this point, growth is remaining constant because the amount of glucose that's being hydrolyzed is staying constant. And there we have it. Let's have a look at the mark scheme, see if we've got the marks. So we've linked sucrose hydrolysis to growth. We've said that as you get more sucrose hydrolysis, that's showing that the plants are growing, but only up to this point. And then after this point, the growth is plateauing, it's remaining the same.